Hi guys, this is Ms. Carlson again. Today we're going to go ahead and take notes on Chapter 7 that covers the discovery of the cell in 7.1, and then in 7.2 it breaks down each organelle and its function that help the cell do the job that it's supposed to do. So before we begin, I want you to make sure you can start off with taking Cornell notes in section 7.1. Once I jump into 7.2 and start talking about the organelles, I put a document that you can print off or get from me in class on the Schoology page. And that will be very helpful to you, and I will reference that document as I go through that section. Okay, so 7.1 is an introduction into how we discovered cells. And basically, it's the result of the invention of the microscope that has greatly influenced cellular biology, or the study of cells. Van Leeuwenhoek used the first microscope to identify organisms in pond water, and then Robert Hooke used a light microscope to look at cork. Cork is a type of plant tissue, and those tissues seem to be made of tiny little chambers, and he decided that those chambers looked like little cells, kind of like jail cells, with their walls, and that's how we got the word or the name for cells. The cell theory was then devised by Schleiden, Schwann, and Vacuoles, and that theory is listed in these 3 bullet points. All living things are composed of cells. Cells are the basic units of structure and function in living things, and then new cells come from existing cells. So you really want to make sure that you can kind of recognize and verbally say these 3 points of the cell theory. The basic structures that are pretty much seen in all cell types are the cell membrane, which is just a thin, flexible barrier around the cell. The cell wall is primarily only in plant cells and bacterial cells or prokaryotes, and that is a strong layer to support that cell membrane, and it is on the outside of those cell types. The nucleus is where the cell holds its genetic material, or its DNA, and controls the cell's activities. And then the cytoplasm is a material inside the cell membrane that basically all the other structures or organelles are kind of suspended in. Now, like I said, all of these structures are the very basic structures that are in most cells, but not in all of them. So make sure you're paying particular attention when we move into 7.2 and talk about the organelles and where they're located. But there are 2 cell types, which also determine if those basic structures are there or not. Prokaryotic cells include bacteria, basically, and they do not have a nucleus, but they do have DNA that's kind of suspended within the cell, and they do have a few organelles in common with some of the eukaryotic cells, which are plant and animal cells. Eukaryotic cells have a nucleus and contain many organelles. Keep in mind, one way to remember this is eukaryotic is talking about you. You are made up of eukaryotic cells, and you are an animal, so that might help you remember that plant and animal cells are eukaryotic. And here is a picture showing you roughly the general difference. Prokaryotic cells are very simple, whereas eukaryotic cells are usually larger and more complex and have multiple organelles, and you can obviously see a nucleus in the eukaryotic cell, whereas in the prokaryotic there is not a defined nucleus. Okay, now is the time where you can get out that 7.2 organelle note sheet and just take your notes on that. There is a picture for every organelle, and I will tell you which letter is pointing to that organelle in the notes. So the cell wall, again, is located just outside the cell membrane, but you're only going to find it in plants, algae, fungi, and prokaryotes. It's made up of carbohydrate fibers and cellulose in plants, and the function is simply support and protection. This is labeled by letter G on your handout. Okay, so the cell membrane is just inside the cell wall of the outermost layer of plant cells, or it's simply the only layer in the animal cell. Now, it is also, like I said, in prokaryotic cells, so it would be right inside the cell wall for them too, but all eukaryotic cells and all prokaryotic cells have a cell membrane, so basically all cells. Now, the composition and structure is made up of lipids, which we will get into more later on, and the main function is for support and protection, and of course regulating things that can enter and leave the cell. This is indicated on the picture by number 5 on that animal cell. Now the nucleus is within the cytoplasm, and it's in all eukaryotic cells. The composition and structure is made up of a nucleolus, which is a dense region within the nucleus that manufactures ribosomes. It kind of looks like a smaller ball within the nucleus, and it is labeled number 11, okay, on that picture on your notes. The nuclear envelope is a porous double membrane that is surrounding the nucleus, so sometimes it's also called the nuclear membrane, and that is identified with the same number that the nucleus is. You can kind of see the holes in it. It's number 2. And then chromatin is the DNA that is bound to proteins, and the chromosomes are essentially referencing the same thing, but your DNA, when it is condensed, we refer to them as chromosomes. Now the function of the nucleus is it's the overall cellular control panel, so it tells everything else what to do. And then of course houses the genetic information, or the DNA. Here is a picture. Again, it's a different picture, but you can kind of look at on your note-taking sheet that the nuclear membrane looks very similar. The nucleolus looks a little flatter, obviously, because this is two-dimensional, but it would be that center region within the nucleus, which again produces ribosomes. All right, moving on to the cytoskeleton, which cannot be seen in the cell picture. It is just free-floating in the cytoplasm, and it is made up of microtubules and microfilaments. These come into play more so when we get into mitosis and the division of the cell, but they are hollow tubes of proteins that kind of move organelles around, and then the filaments are long fibers that are going to be supporting the cell and play the greater role in cell division. So their job is to maintain cell shape, form cilia and flagella, which are tiny little tentacle type objects for movement, and then transport organelles and chromosomes. And in this picture here, you can kind of see a cross-section of a cell, and here are the microtubules, and the microfilaments are a little bit finer. All right, ribosomes. They are identified by number 1 in the picture on your handout. They are also within the cytoplasm and can be found on other organelles, specifically the rough endoplasmic reticulum. They are made up of RNA and protein, and they are very small bead-like structures, so they look like little sprinkles kind of within the cell. They aid in protein synthesis, and to synthesize means to create, so basically they make proteins. Very important organelle. Then the endoplasmic reticulum that I just mentioned is within the cytoplasm, very close to the nucleus. And that's how you can differentiate it from the Golgi complex that we'll see here shortly. But it is an internal membrane network, so it kind of looks like a series of folds, or kind of like if you think of a fruit roll-up all folded together. The two types are the rough, which is just rough because it's studded with ribosomes, kind of giving it that rough texture. And then the smooth ER is free of ribosomes, but essentially looks the same with those folds. The rough ER is going to help with the modification and transportation of proteins, and then our ribosomes specifically, which are making the proteins. And then the smooth ER is going to help assemble cell membrane components, which we remember are known as lipids. And let's see, in your pictures, the smooth endoplasmic reticulum is identified by number 8, and the rough is identified by number 3. And again, here's a little close-up of the rough endoplasmic reticulum with the ribosome studded on it. Again, the smooth would look very similar, just without the ribosomes. Now, the Golgi complex is also called the Golgi apparatus, or the Golgi body, also suspended within the cytoplasm. Again, another series of folds and sac-like structures. It kind of looks like a stack of pancakes, but it's not going to be close to the nucleus like the ER is going to be. The function is for them to package and transport proteins to other parts of the cell. They also play a role in modification. And on your picture, it is located on number 4, or it is identified by number 4. Here is a close-up. And again, it looks similar to your picture. All diagrams are going to look a little bit different, so just you got to use everything and your knowledge about that organelle to make sure you're able to identify it in a picture. Now, the lysosomes are within the cytoplasm. They are small organelles, and they are composed of enzymes to help break down lipids, carbohydrates, and proteins for use by the cell. They also help in breaking down old or broken organelles. So they're kind of like the trash cans of the cell, if you will. They are located or being labeled by number 7. Now the vacuoles, again, just suspended in that cytoplasm, they are also sac-like structures and appear as kind of like open spaces. They're very small in the animal cell, almost look like the lysosomes, but in a plant cell they're very easily identified because they are very large and there's only 1 to 2 of them. And for plants and animals, they're going to store water, salts, proteins, and carbohydrates. And then also in plants, so the pressure within the vacuole actually helps maintain the cell structure. And that is labeled by a letter E on the plant cell picture for that organelle. Chloroplasts then are only going to be found in a plant cell. They are within the cytoplasm, and they have their own membrane and DNA. So this is very important because chloroplast is one of the 2 organelles that are membrane bound and have DNA. They contain chlorophyll, which is what allows the plant cells to capture sunlight and enable for it to produce food in the process of photosynthesis. And you will find the chloroplast identified with, I believe, that is letter H. And here's a close-up. We will talk in more detail about the inner structure of the chloroplast, but for now, really you just need to be able to identify it and know that it's only found in plants. The mitochondria within the cytoplasm and in all eukaryotic cells. Many people make the mistake of thinking that they are only in animal cells, but they are also in plant cells. And if you want to circle letter L in the previous plant cell for the chloroplast, that is pointing to the mitochondria there. And in the animal cell that is actually in the row for mitochondria is number 9 is what you will need to circle there. Like I told you before when we were talking about the chloroplast, it's the 2nd membrane-bound organelle and has its own DNA. And its function is to use energy from stored food molecules to aid in growth, development, and movement. Remember, these are usually identified as the powerhouses of the cell because they help to power the organism itself. And here's a close-up. A lot of people also like to say that the mitochondria looks kind of like a hot dog with, you know, mustard or ketchup, whatever you like to squirt on it. And that is our last organelle. This is just a picture showing you a plant cell. And I know it's a plant cell because the vacuole is very large. It has 2 outer barriers, one being the cell wall and the inner one being the cell membrane. And of course it has chloroplasts. And then here you have an animal cell. Only has 1 outer barrier, so I can tell right away that it's an animal cell. And that is its cell membrane, that outer barrier. And we are pretty much done. We will talk a little bit more about this in class and start working on identifying these organelles in pictures because they all look a little bit different. As you can see, this animal cell looks different than the one in your notes. But all models are different, but all of them can be identified if you know where those organelles are generally located and their characteristics. So have a good day, and I will see you next time.